Every moment we're awake, our brains make sense of what we see, but your brain is not like a camera. It constantly decides what information to keep, and even the most advanced AI systems struggle with this.
Understanding how the brain does this is a central question in neuroscience. Our work focuses on how individual neurons in primary visual system contribute to this process.
Now, for decades, visual neuroscience has explained neuronal responses using human-defined features and categories like edges or textures or specific classes of objects like faces. And that framework works well in controlled settings, but it sort of begins to fail when neurons are tested with the enormous diversity of images that is found in the natural world.
In areas like inferior temporal cortexes, a single neuron can respond strongly to images that looks completely unrelated to each other. A truck, a centipede, or a bridge can all drive the same neuron.
To us, those images have nothing in common, but the neuronal response is consistent and reliable. That tells us that the neuron is responding to something meaningful that our perception does not naturally highlight.
So the core question became, what are these neurons actually encoding? We suspected that they are responding to local visual features that repeat across very different images.
The challenge is that humans are not very good at isolating that kind of feature, that kind of local feature, because we naturally focus on whole objects. So this project was about finding a way to let the neurons show us what really matters to them without forcing our own assumptions onto them.
We took a bottom-up approach. Instead of starting with a narrow hypothesis, we let the neurons themselves drive the experiment.
We use closed-loop image synthesis, where the neuron's own firing rate guides the generation of new images in real time. Because every generative model also has an intrinsic bias. Different models kind of carve up visual space in very different ways.
So instead of trusting a single model, in the work, we deliberately used two very different ones. One model will control texture-like structures.
The other model emphasized more object-like, photorealistic objects. We ran both models in parallel during the same recording session.
To understand this setting, we can consider the following metaphor.
We can recruit two painters with very different artistic styles to work for a patron. You can imagine that this painter, whose style is better aligned with the patron's taste, can more readily produce a painting that satisfies her. We use this notion, which we're calling alignment, as the ease in which the preference can be communicated and realized through interaction from the patron, the neuron, to the painter, the model. And that's the object of study.
In that first experiment, we run two evolutions with the two generators in parallel, one in each model. And the images looks totally different globally.
But in both evolutions, the same eye-like pinkish local feature emerged in basically the same spatial location.
The neurons are pulling both models towards the same local features,
even though the models expressed it in completely different visual languages.
The alignment between neurons and the models changes across the visual hierarchy. Neurons in early and mid-level visual areas align better with texture space and have a harder time guiding evolution in the object space. Neurons in posterior inferior temporal cortex align comparably well with both texture and object space. Together, these results suggest that neurons encode flexible local features that can be recombined into objects, rather than rigid object templates.
So this work would suggest that artificial intelligence models are not just tools for modeling perception, but tools for revealing what biological neurons care about. So in some cases, artificial intelligence models may be necessary to see past our own perceptual biases.